right, everybody, it's 12:30, so let's get started. And just before we officially start, I just want a couple of reminders. So if you haven't already submitted your project topic, they are due tonight. So please submit your project topics before 11:55 tonight if you haven't already. A bunch of you have come to me about them, which is awesome. If you haven't, please send me something through Brightspace. Submit through Brightspace. And the other thing, as you hopefully know, is your exam is coming up on Thursday. So maybe not everyone's favorite use of class time, but that's what we'll be doing on Thursday in this room from 12:30 to 2:10. If you need extra time or other accommodations, please talk to the ODR about that. And for the exam, make sure you have a pen or pencil and a calculator. You'll be given a reference sheet, which is already posted on Brightspace with all the references you'll be given. And if you need to do calculations or show your work, please do so just right on the exam. So don't bring extra scrap paper or anything like that. Just pen or pencil and a calculator. Yeah. Okay. Yeah. It's on the test, just on the paper. Yeah. Any other questions about the exam before we get started? I believe there are 27 questions. I saw faces in the audience. Yeah, so there's some multiple choice, some fill in the blank, some short answer style where you write a few sentences, some drawing, I think there's some matching, a little bit of everything. Yeah, and yeah, exactly. So today we're going to finish going over chapter 5, and then if anyone has any review questions they want to go over from anything we've learned so far this semester, bring it up and we'll go through it. And if you need to talk to me just individually, I have office hours after class today. You can make an appointment with me tomorrow if you want to see me tomorrow. And on Wednesday, I'm extending my office hours, so they'll be from 1 to 3. And there won't be office hours Thursday this week because none of you probably want to talk to me after you're finished with that long exam. You can make faces at me next week. So, yeah, so I think that's all the housekeeping reminders I have for you. So let's finish up with chapter 5. So last week on Thursday, we finished going over this question, which was comparing the different techniques for measuring proteins. And just as a review, we noticed that some of these techniques are really good at pulling out just your protein of interest from a mixture of proteins. And those are going to be ELISA. Oops, forgot to turn my pen on again. Those are going to be ELISA, detects only your protein of interest very specifically. The activity assay measures the ability of your protein of interest to catalyze a reaction, so it's very specific for your protein of interest. These other methods aren't able to differentiate your protein from a mixture of proteins, right? Just measuring absorbance at 180 or at 280. All proteins that have tyrosine and tryptophan are going to absorb at 280. You can't tell the difference between proteins just looking at absorbance. Same thing with the Bradford. All proteins are going to take on the Bradford reagent, and it will turn color. It doesn't matter what protein it is. It likes all the proteins. Gel electrophoresis, same idea. It uses that same Kumasi dye. It sticks to all proteins. Gel electrophoresis helps you to see how many different proteins you have, but it doesn't tell you anything special. It doesn't just pick one out over the other, right? It detects all of them. Any questions about these techniques? Okay, so let's move on to the next question. So we have 3 different samples. We did an activity assay, right? We measured units for our protein of interest. So activity assays measure enzyme activity in units, which is a sort of an arbitrary measurement of enzyme activity. And so for each of these samples, we measured how much total protein we have, maybe with a Bradford assay, and then how many units of our protein of interest using an activity assay. So which of these 3 samples has the most pure protein? So everybody take a few minutes and think about that one. All right, does anybody want to tell me which sample is the most pure for our protein of interest? You have a 1/3 chance of being right. Yeah. Yeah, it's sample C, exactly. How do you know that? Mm-hmm. Yeah, perfect, exactly. So when we're trying to figure out which sample is the most pure, when we have information about units of active protein and total protein, we want to look for what we call the specific activity. So the specific activity measures how much activity in units per total protein, which is in milligrams. So if you do this fraction, right, you get how many units per milligram of protein. So the most pure protein would have the most units for every milligram of protein. Does that logic kind of make sense for everyone? So for sample A, we would want to divide our units by our milligrams. So we have 0.3 units over 0.4 milligram. We'd have 0.75. For sample B, we would be dividing 0.1 divided by 0.3, which gives us about 1/3. And for our last one, we have 0.2 units divided by 0.1 milligram, which would give us 2. So 2 is by far the largest number here, so that makes sample C our most pure protein. Any questions about that? Okay. So now we'll move on to the next, I think it's the last section of chapter 5, which is the further characterization methods. So we'll start by talking a little bit about ESI-MS. So first of all, what does ESI-MS stand for? It's a long one. If you just know part of it, you can tell me part of it also. Yeah. Yes, the MS is for mass spectrometry. Help if I could spell that. Does anybody remember what the ESI stands for? Yeah, the S is for spray. All right, so the ES is electrospray. And the I is ionization, which basically describes what we're doing, right? We're sort of spraying these proteins out to try to separate them all, and we're doing so based on charge. So we're ionizing them. And we're using this electricity, like voltage difference, to charge all the proteins and repel them from one another. So you have a sort of, for lack of better words, kind of like a spray. So yeah, so the proteins are separated. Separated by their charge, and also because they're in a vacuum. And then once we do that electrospray ionization, then we can use our mass spectrometer to actually measure the apparent something of a protein, which is our next blank. So what does ESI-MS directly measure? What property of a protein? Yeah, charge is part of it. So if you remember what that little spectrogram, like the graph, looks like, what's on our axis that we're actually measuring? Yeah. Yeah, exactly, the mass per charge ratio. So yeah, we can directly measure this m over z. And so because we're talking about adding and subtracting protons when we're changing the charge, every time we change the charge, we also change the mass a little bit because protons have a little bit of mass. So when we measure the mass per charge ratio, we don't get the exact mass of a protein or the exact charge, but we get this ratio, which we can then pull apart using an equation to figure out the blank of a protein. So what from those peaks, from those m per z measurements, what can we determine about the protein? Yeah. Yeah, exactly. The molar mass or the molecular weight. Any questions about these kind of basics of what ESI-MS is or what it does? Okay, so let's do a practice problem. So here is a typical spectrum that you'd see from an ESI-MS assay. So first of all, on the spectrum, what are all the peaks that we're looking at? What are they? What are we looking at? Are we looking at a bunch of different things? Are we looking at one thing in a bunch of different ways? Yeah. I mean, like, so you have all these different peaks here. Does each peak, what does each peak like represent? Yes, I mean, it represents a mass per charge ratio, right? But so if you're measuring just, you put one protein into your ESI-MS machine, and you're getting out this, how do we have one protein that can give us all these different peaks, right? Why are we seeing all these different peaks, even if we have one pure protein here? So, not quite. So, all right, yeah. Exactly. Yeah. So like the more protons you take on, the more charge you take on, and then and each one will affect the mass very slightly. And so that's why each of these peaks is labeled with the charge and also the mass per charge, the m per z ratio that's measured. So if you know the charge and you know the m over z, you can figure out the original molar mass. But each of these peaks actually represents the same protein, and you're just looking at the relative abundance for each. So like your one protein, most of it, you're measuring this peak, the plus 19 peak, that gave you that particular m over z. But this represents actually the same protein. Just you happen to measure less of it. And it has a different mass per charge ratio because the charge now is only plus 11. So, so as we go through this problem, we can kind of see how that works. So how can we figure out the molecular weight of the protein that's represented by the spectrum? Yeah. Yeah, exactly. So this is the formula. So P represents the peak that you measure, right? This is your apparent mass per charge ratio. So like this would be your P. And then your Z is the charge that's measured, right? That's also given. So here's your charge. And then M is what we're trying to find, right? M is the actual molecular weight of our protein. So if everybody can take a few minutes, everyone can figure out what they think the molecular weight of this protein is. All right, so does anybody have an answer for what is the molar mass of this protein? Yeah. Yeah, 16,950 what? 4. Awesome. Did anybody get, and which peak did you calculate that from? Cool. So you got 16,954. Did anybody else get a slightly different molar mass using a different peak? Yeah. Okay. Which peak was that from? 11. Okay. 16,951. Anybody else use a different peak? Get a similar or different molar mass? Yeah. For the plus 10? 16,979, you said? For plus 19, sorry. All right. So, yeah, so like all of these, hopefully this kind of shows you that all these peaks are measuring the same protein, just it has taken on different amounts of charge. And so different, the protein apparently really likes to take on 19 protons. It likes to take on 10 proteins less because there's lower abundance of that. But those are all just different representations of what the protein is like when it's taken on different amounts of charge in this ESI-MS apparatus. So, and does anybody need to go over that math? So when you solve for M, you rearrange this equation. So you get M equals PZ minus Z, and then you can just plug in whatever P and Z number you choose from the graph, and each one will give you something slightly different. And so this can be a really accurate measurement because so even though each one of these peaks gives you, a slightly different number, a computer can statistically average them all together based on their abundances and get a really exact calculation for the molecular weight of a protein. And from there, you can potentially help go on to identify what it is. So the other thing I did not ask you is when you gave an answer for the molar mass, what units is it in? Yeah, so usually when we give molar weights, it's in grams per mole. Has anyone ever learned another unit name that is also in grams per mole, but is a fancier name for it? What's that? You can use that. I guess it's more of like a chemistry thing. I'm thinking of a different one that's usually used for like proteins specifically. Yeah. Daltons. Yeah, exactly. So another word or another term for grams per mole is Daltons. That's often abbreviated DA with a capital D. Daltons. A lot of times, since proteins are so big, we'll give them in kilodaltons. So in this case, you would have 16.9 or so kilodaltons. I guess it would really round to 17.0 kilodaltons, but neither here nor there. But yes, yeah. Yeah, sure. So if we wanted to calculate the molar mass using this equation, let's use let's use this one, the plus 14 and the 1211.8. So our molar mass would be P, which is 1211.8, times Z, which is 14. And then you just subtract out Z again, which is 14. And I don't actually have a calculator on me, but that should give you about 16,900 or so. 16,951. That's good enough. Yeah, cool. Thank you. So, any other questions about mass spec, reading spectra like this, calculating molecular weights? Okay. So let's go into the primary structure of a protein. So can anybody tell me what is the definition of the primary structure of a protein? Yeah. Exactly, yeah. It's the amino acid sequence. In a polypeptide. So if we have a protein that is 50 amino acids long, there are how many possible amino acid sequences? Yeah. Yes, it is 20 to the 50. How do you know that? Exactly, yeah. So at each position in the sequence, there are 20 possible ones. So you have 20 times 20 times 20 times 20 for as long as your sequence is. So in this case, if you have 50 amino acids, it would be 20 to the 50th power. And so just as a sort of reminder, or compare contrast with something like DNA and RNA. DNA and RNA are nucleic acids. They're great. You can get a wide variety in them, but they only have 4 different options at each position. Amino acids, or proteins, on the other hand, have 20 options for each position. So this gives you an enormous possibility for variety in terms of primary structure, which then leads into secondary structure, tertiary structure, function, all that stuff. So you can, even though there's only 20 amino acids, you can make this massive variety of proteins that make up a lot of life on Earth and all that good stuff. Any questions about this? Okay. So now we will get more into protein sequencing. So let's talk about endopeptidases. What reaction do endopeptidases catalyze? What are they doing to a protein? Yeah. Yeah, exactly. Yeah, so they're breaking peptide bonds in the middle of a peptide, or just not at the ends. So you could say you're breaking a bond, you're cutting the bond, you're cleaving the bond. Does anyone have a more specific word for what we're doing to cut that bond? Are we adding a molecule? What? Yeah, exactly. We're doing hydrolysis. So we are using water to cut our bond with the help of an endopeptidase enzyme. And we already know that, or I sort of hinted at, this is helpful for sequencing, which we'll get to in a little bit. So, yeah, so next part of the question, endopeptidases are a type of what? Yeah, they're a protease, exactly. They help to cut those peptide bonds that are in proteins. That makes them a protease. So just as a little review or a compare and contrast with DNA again. So we have, for part A, we have restriction enzymes. Those also cut stuff, right? How are those different from proteases? What do restriction enzymes cut? Yeah, restriction enzymes cut DNA. Proteases cut proteins. They both, but yeah, they're both yeah, I guess only one of them is really used for sequencing. But yeah, make sure that you know the difference between the targets of a restriction enzyme, which targets DNA, and a protease, which targets protein. All right, so let's think about proteases. Does chymotrypsin cleave the following peptide? And if so, where? So this here is the peptide we want to cleave. And then on the bottom here, we have part of that table that has our list of proteases and where they cut. And this little picture just shows you which peptide bond is being cut and what the definitions between Rn-1 and Rn. So Rn-1 is the residue right before where you're cutting. Rn is the side chain of the residue right after you're cutting. So does chymotrypsin cleave this peptide? You take a few minutes and look at our definitions and decide. All right, so does chymotrypsin cut this thing? Yes or no? Yeah. Yeah, where does it cut it? Yeah, exactly. So it's going to cut right here. And how do you know that? Mm-hmm. Yeah, exactly. So right before the cut, we're looking for Rn-1. So according to our table, this thing, chymotrypsin, is going to cut right after it sees a bulky hydrophobic residue, and those are phenylalanine, tryptophan, and tyrosine. So we have a phenylalanine right here, so that can be our R-1 or Rn-1. And then the only stipulation we have is that our Rn, the amino acid right after, cannot be a proline. And a lot of proteases have this stipulation. They don't like to cut right before proline. And we talked a little bit about why. So like proline is going to force the amino acid bonds to take on a particular angle because proline has that extra little sort of loop as part of the side chain. And so anytime you have a proline, it's going to limit the shape and the ability of a protein to fit into the active site of a protease like this. So if you need the protease needs to get physical access to the spot where it wants to cleave, and a lot of time proline just physically prevents this from happening. Any questions about proteases? Yeah. It would probably do a mixture. So unless it's not like these three are given a priority, but it is also an endopeptidase. So once it cuts after one, the other one's on the end, so it wouldn't cut after the other one, right? So probably just a mixture, or it could depend on the exact shape of the protein. Maybe one of those side chains is more available than the other one, or because of just the actual folding of the protein. So there's no, I don't think, a rule of if you had a choice of two right next to each other, which one you'd pick. It would be a mixture. Yeah, yeah. Yeah, the longer you let the chymotrypsin go, the more it's going to get. Yeah. Yeah. Yeah, so if that's the case, it would definitely depend on how long you let it react. So first, if you stop the reaction fairly quickly, you would see mostly cuts after those hydrophobic residues. But if you let the reaction go on for a long time, then you would start to also see cuts after those other bulky-ish residues. Someone would have to tell you, yeah. I mean, I will say for the purposes of the exam, you won't have to worry about these sort of secondary cut sites. And I think in the actual reference sheet on the exam, only the primary cut sites are even on there. Like this isn't there, so you won't have to worry about that in terms of the exam. But it's something, I guess, useful to know if you're doing it in the lab. Exactly, yeah, it would cut after any or all of them, depending on exactly where they are in relation to each other. Yeah. Yeah. No, so yeah, usually it's just always in relation to the bond that's being cut. So the bond that's being cut can change depending on which bond is being cut, but it's always just right before and right after, n-1 and n. For whatever reason, they're numbered that way. Okay, so let's now talk a little bit more about protein sequencing. So how do we accomplish protein sequencing? Is it by sequential addition of complementary residues, sequential removal of N-terminal residues, or the hybridization of two polypeptides? Yeah. Yeah, exactly. So this is called Edman degradation. We'll review the steps for that in a few minutes in case anyone forgot. But yeah, so what we do for Edman degradation is one at a time. We take away one amino acid, always the one from the N-terminal end, and you can identify what each amino acid is one at a time. So it's a relatively slow process because you had to kind of look at each amino acid one at a time. Okay, so let's think about Edman degradation. So everybody take a couple minutes and put the steps in order. All right, can somebody tell me the first step for Edman degradation? Yeah, B. Yeah, so the first thing we do is we functionalize the amino terminal amino acid with PITC. What is the next thing that we do? Yeah. Not quite. There's something before there. Yeah. D. Yeah. So after we functionalize the N-terminal end, we then cyclize it and remove it. And then once it's removed, what do we have? Yeah. Yeah, so once it's removed, we have this PTH amino acid that's been separated from the rest of the polypeptide. So then finally, the last thing we do, the last one left obviously, is we're going to actually identify what amino acid we have, usually by chromatography. So just as a recap of that, let's look at our slide. So first we add PITC, which is up here in red. We have our N-terminal amino acid in blue. we have this cyclization process that happens through the addition of trifluoroacetic acid. And we get this PTH amino acid eventually that separates out, which we can then use chromatography, like a column or even thin layer chromatography, to identify what we got. Yeah. Not like the structure, like I won't ask you to like draw the structures or anything, but know what the steps are and the order they go in, and like basically, yeah, that should be fine. That should be fine. Yeah. No, not in terms of like electron pushing. No. Yeah. Any other questions about Edmond degradation? All right, so we can use Edmond degradation to then put together sequences for larger protein fragments. So we have here a practice question. So we have a peptide. We separately cleaved it. Some of it we cleaved with cyanogen bromide. Some of it we cleaved with chymotrypsin. And we got these different fragments. So everybody take a few minutes, and I'll give you some more information first, and then I'll ask you to take a few minutes to identify what the intact peptide sequence is. One other really important thing to know before you start is where each of these things cuts, right? So cyanogen bromide cuts after methionine. Chymotrypsin cuts, like we just saw, after our bulky hydrophobics. So now that you know that, that should be enough information for you to start putting the fragments in order. So I'll give everybody a few minutes. Alright, it looks like most of you are done, maybe. So, can anybody tell me, how did they start? What's the first thing you did to try to figure this out? Yeah. Mhm. Uh-huh. Uh-huh. Okay. Yep. So what does that mean about the first one? Yeah, exactly. So this one has to be at the end. Okay. So yeah, that's a really good start to figure out which one is last. Uh-huh. Yeah, that is correct. But yeah, first let's walk through a little bit more about how we solved that for anyone who wasn't sure about how to get there. So a really good approach is to figure out which one is last. So for cyanogen bromide, fragment number 1 is last. For chymotrypsin, which fragment has to be last? Yeah, the 6th fragment. Yeah, so this one is also last. Did anybody start from the other way? Did anybody figure out which one was first and go forwards? Or did everybody start from the end and go backwards? So sometimes, not always, you can figure out which one the first one is, because the beginning end of either fragment is going to be in common between the two methods, right? So it doesn't matter where they cut, the first amino acid is the first amino acid. So in this case, the only one that's first that they share is aspartate. So you know that aspartate has to be first because it's shared between the two cutting methods. Does that make sense to everyone? So this doesn't always work out, right? Sometimes there's more than one shared first one, but in this case, that can also help you figure out which one is first. Because that's oh no, it's the same protein just cut in 2 different methods. Yeah. Okay, sorry. Yeah, sorry if that was not clear. Yep. Okay. So let's go over a little bit how I approach this one. So knowing which one is first is nice as a sort of check, but you can't always tell. So a reliable way to do it is always to start from the one at the end. So we already talked about how we know that fragment 1 has to be last for our CNVR because it doesn't end with a met. You didn't cut after it. Fragment 6 has to be last for our chymotrypsin cleavage because chymotrypsin doesn't cut after asparagine and it cuts after the other 2. So it must have cut after 4 and 5. It didn't cut after 6. So once we have fragment 1 and fragment 6 at the end of each of our peptides, we can start filling in, right? So we know glycine and asparagine, and both of them end with there, but in our fragment 1, we have some additional information, arginine, alanine, and tyrosine. So what does that tell us about what has to come right before fragment 6? Which fragment has to come right before fragment 6 now, out of 4 and 5? Yeah. Exactly, yeah. So the one that has a tyrosine there has to go right before that so that the fragments will continue to match up. And then we can similarly go through that again. So now we have the matched up sequences up to the arginine, but we know our fragment 4 has a methionine. So does that help us know which one of fragment 2 or 3 we can put at our top polypeptide? Not really, yeah, because they both end in met, right? So in this case, this is no longer helpful for us, but luckily we only have 3 fragments, so we know that fragment 5 has to come right before fragment 4, right? And that will give us the full sequence. And so from there, we can then fill in the ones at the top. So you can sort of take you have the same polypeptide fragmented 2 different ways, and you can use, because the different fragmentation methods are in different locations on the peptide, you can then line up your fragments in this way, either from the end to the beginning or sometimes from the beginning to the end, and figure out what you got. Yeah. So there's 2 reasons. So the first thing is that the only fragment that's shared as the first fragment in both of these, or the first amino acid residue, is aspartate. So you know that the other fragments can't be very first because they'd have to share the very first amino acid residue in common. They're the same, yeah. Yeah, so this is the concept behind protein sequencing, is you have the same protein, you have 2 copies of it, one of it you fragment one way, one of it you fragment a different way, and then you can align them to get the orders. Once you've done Edmond degradation and you figure out what amino acid is in what order, right, for each fragment. Any other questions about that? Okay. So let's define some relationships. So this is the ways that proteins can be related to each other. They can be orthologs, homologs, or paralogs. And those describe different ways that proteins can be similar to each other. So for question 1, what would we call any 2 proteins that share a common ancestral gene? Yeah. Yeah, this would be homologs. So homolog is the broadest term we have here. It refers to just any 2 proteins that share a common ancestral gene, regardless of whether they're in the same or different organisms, or whether they have the same or different function. It just relates to proteins that share a common ancestor. So for number 2, 2 proteins that share a common ancestral gene, but after gene duplication underwent divergent evolution and now carry out different functions in the same organism. Yeah. Paralogs, yep. So paralogs are a type of homolog, right? They share a common ancestral gene, but something special about paralogs is they result from a gene duplication, and they refer to proteins that after gene duplication and evolution have 2 different functions. And that can be in the same organism, and it can sometimes be in a different organism. But the idea is common ancestor, different functions. So then we have number 3, 2 proteins that share a common ancestral gene and carry out the same function in different organisms. Got 1 choice left. Yeah, it is A. So orthologs are a type of homolog, right? They share a common ancestral gene, but orthologs carry out the same function in different organisms. Any questions about these relationships between proteins? All right. So we have 2 proteins, hemoglobin and myoglobin. They are derived from a common genetic ancestor. Hemoglobin carries oxygen in the blood, and myoglobin binds oxygen in muscle tissues. So first, let's compare human myoglobin and human hemoglobin. Are they homologous, orthologous, and or paralogous? Yeah. Yeah, they're paralogous. Are they anything else? They're also homologous. Yeah. So what makes them paralogous? Uh-huh. Mm-hmm. Exactly, yeah. So they have different functions. They have a common ancestor. And they're now in the same organism. So let's now think about human myoglobin and whale myoglobin. Are those going to be homologous, orthologous, and or paralogous? Yeah. Yeah, so these ones are going to be orthologous. Anything else? Also homologous. Yeah, so what makes them orthologous? Exactly, yeah. Common ancestor. Same function, or similar function. Different organisms. All right. Any questions about those fun new vocab words? Yeah.
It looks like here, like
Yeah, I mean, if it's the protein, if it has the same name and it does the same thing, it's probably a homolog, and it's probably an ortholog if it's in two different animals or two different organisms. Yeah. Any other questions? All right. So let's think about mutations. So we're going to take a few minutes and classify each mutation as one thing from each of these rows. So first I'll explain the rows and then I'll explain these pictures because they're maybe a little confusing. So you want to select one of A through E. So like what type of mutation is it in terms of frame shift, point, insertion, deletion, or none. Then you should also choose from the second row, is it missense, nonsense, silent, or none? And then choose from the final row, is it conservative, non-conservative, or none? And so this is, on the left, we have our original codon, and then our original one is going to turn into each of these other four. So we have four different questions. The original to this one, the original to that one, the original to this one, and the original to the final one. Does that make, does everybody understand what the question is asking before we get started? Do you have a question before we get started? Or you wanted to answer one? That's a very good question, and the answer is you can't tell. Yeah, so this isn't a great example for looking for frame shift mutations, insertions, and deletions, because you only know what's happening in this particular codon. So you don't know if everything got moved over or if it just got, changed. But do your best. If I give you something like this on the exam, there will be more codons. So everybody take a few minutes. right, so for this first mutation over here, does anybody want to tell me the way they classified it with each of those three things? Yeah. Yeah, they pretty much, from what you can tell, they look like point mutations. So yeah, you can start by saying this one is a point mutation. What else? Yeah, it is silent. So what do you mean by silent? What makes a mutation a silent mutation? Exactly, yeah. So our original codon coded for lysine. Our mutated codon still codes for lysine. It is silent. So is it conservative, non-conservative, or none? So, okay. So usually when we are talking about conservative and non-conservative, we're talking about when an amino acid is actually substituted. In this case, there is no change in amino acid, so it is none. Yeah, there's no change, so it's not conservative or non-conservative. It's just the same. So like if it changed from lysine, which is basic, into another basic amino acid like arginine, which I'm giving away now, it would be conservative, right? If it stays lysine, it never changed, so it's not conservative and it's not non-conservative. It's just itself. Okay, so does anybody want to tell me about our next one here that says ATC? What type of mutation is it? Yeah. Yeah, it's a point mutation. It is a nonsense mutation. So what does it mean to be a nonsense mutation? Exactly. Yeah, a nonsense mutation changes our code from an amino acid into a stop codon. And what about our last set of criteria? It's none of the above. Yeah, so you're not changing into another amino acid, so it's not conservative or non-conservative. It's just nothing. All right. Anyone want to do our next one, which says TCC? Yeah. Yeah, this is also a point mutation. It is missense. And so what does it mean to be missense? Exactly. Yeah, so since we changed one amino acid into another amino acid, we finally have something to say for our third classification, which is what? Yeah, it's conservative. So what makes it conservative? Exactly. Yeah, they're both basic. So lysine, so the coding is sort of up here. So lysine is basic, it's purple. Arginine is still purple, still basic. Same type of amino acid, conservative mutation or conservative substitution. Okay, so what about our final one that says TCG? Yeah. Okay. Exactly, yeah. So what makes it non-conservative? Exactly, yeah. So instead of being positively charged and basic, now it's just kind of polar. So that could potentially change how this amino acid residue interacts with other stuff, whether it's other parts of the same protein or other proteins or whatever its job is. It might affect how it does its job. Does anybody have any questions about classifying mutations? Okay. So let's talk a little bit more about substitutions. Oops. Computer is grumpy. All right. So let's talk about substitutions. So we have amino acids on the left over here, and I want to know which amino acid over here on the right is the most conservative substitution for each one of these on the left. So everybody take a couple of minutes to think about that, and then we'll go over it together. Alright, so to know whether or not our substitutions for our amino acids are conservative or non-conservative, first, of course, we have to be able to identify the side chain of each amino acid, because if the R group or the side chain is similar, then that's a more conservative substitution than one that's dissimilar. So if you weren't sure how to approach this problem, the first thing you might want to do is just look through all of these and decide where is the side chain. And so you know that you have your amino acid backbone with your amino group, your alpha carbon, and your carbonyl group, and the side chain is what comes off of each of our alpha carbons. All right. So once you found all your side chains, let's decide what is the most conservative. So let's start with serine. Which amino acid is the most conservative substitution for serine? Yeah, threonine. Yeah. So serine has a hydrocarbon and a hydroxyl. Threonine has 2 hydrocarbons and a hydroxyl. Very similar in terms of polarity and size. There's just one extra methyl group on the threonine. Okay, so let's talk about aspartic acid. What is the most conservative substitution for aspartic acid? Yeah. Glutamic acid. Yeah, and why is that? Yeah, exactly. So they both have a carboxylic acid as part of their R group. Glutamic acid is just a little bit longer in terms of its hydrocarbon chain, but otherwise very similar in terms of size and properties. They're both going to have the same charge for the most part at similar pHs and be able to interact with other stuff in a very similar way. All right, so let's talk about leucine. What is the most conservative substitution for leucine? Yeah, isoleucine, yeah. And why is that? Exactly, yeah, they're both nonpolar hydrocarbon chains. Again, the difference is just one extra methyl group. So yeah, so things that you want to consider when you're thinking about substitutions are the category of the amino acid, right? So we have our positive and negative ones. So these are our acidic and basic ones. We have our polar and nonpolar ones. And then we also have our large ones and our small ones. So those are sort of three kind of categories you can think about when you're deciding if amino acid side chains are kind of similar to each other or kind of different from each other. Size, charge, electronegativity. Any questions about that? Okay, so we'll do our last couple of practice problem type things on our favorite virus, SARS-CoV-2. So we've been talking a little bit about mutations in DNA and how that can lead to amino acid changes in a protein. And SARS-CoV-2 is pretty good at that. So a lot of the time when we see a new variant, it's often defined by mutations in its genome that have led to changes in the amino acid sequence in some of its proteins. And so, and we saw and then this sort of Venn diagram shows us some similarities and differences in the types of mutations that we see in each of the various major variants that they looked at in this paper. And you can see for Omicron, for BA2, it has a really, really large number of mutations that are distinct from a lot of the other variants, which might help make it more fit, more able to spread. Right now, all it tells you is that it makes protein a little bit different. And so most of the mutations that we'll see are in the spike protein, but we'll also see changes in some other proteins like the nucleocapsid protein. So let's look a little bit more closely at the mutation profile for the spike protein. So this is a different paper, but they looked at the different mutations that were similar and different between the BA4 and BA5 variants, which are also similar to BA2, except for a couple of key differences, which are circled on here. So it's helpful to look at what I want you to get out of this for the test is sort of how to approach looking at mutations, like when you see a description of one. So, like, you don't have to remember, like, oh, there's 25 mutations in spike, but, like, probably useful to know that, like, sure does have a lot of mutations. And what types of mutations are we seeing? So one type of mutation that we're seeing here, so it says delta 69 to 70. So that means there is a deletion in amino acids 69 and 70. So the delta deletion. So good to know, if you see a delta there, that means that whole codon got deleted. So that is a deletion mutation. So another type of terminology, I guess, for writing out a mutation is like we see for these other ones. So this example next that says L452R, so that tells you the position of the amino acid within the protein. So it's amino acid number 452. It tells you what it originally was, which is a leucine. So it changed from a leucine, and it changed to what's written afterwards, which is an arginine. So if you change from leucine to arginine, is that a conservative or a non-conservative mutation? What types of amino acids are they? Is leucine nonpolar, polar, charged, acidic, basic? Yeah, leucine is nonpolar. Yeah, what about arginine? It's basic. Yeah, so if we went from nonpolar to basic, is that conservative, non-conservative? Non-conservative, yeah. So even knowing this sort of very basic information about amino acid changes can give you a clue. So it might be that if this particular mutation is in a location of the protein that's, the active site for an enzyme, for example, or some part of the protein that needs to interact with something else, and suddenly it changed from being uncharged to positively charged, that protein might suddenly behave very differently now than it used to. So that's something that we want to keep in mind when we're looking at sort of real world examples of evolution and protein amino acid changes. Okay, and then the other type of mutation sort of nomenclature that you'll see is like this, the reversion to wild type. So when you see that, you'll just see the original amino acid, which was glutamine, and the position it's at, 493. So what happened here was this glutamine had changed into an arginine in some variants, and it went back to its original. So that's just noted as having the original amino acid there in the front and nothing afterwards, because it went back to the original. Does that kind of make sense? So like it had been Q493R. So at some point in the past, this Q changed to an R. But in this version, in BA4 and 5, the R changed back to a Q. So this is written as just Q493 because it's back to its original. Does that make sense for everyone? Exactly, yeah. So like it changed to an R, maybe that didn't help give it some kind of advantage. And so viruses that had that particular mutation weren't selected for to replicate, and so it got lost over time. Yeah? Sure. Deletion. Like what? I'm actually, my strong point is not like virus phylogeny. I mean, I know that they're sequential, so like we started with BA2, and then over time we have gotten BA4 and BA5, but I could not give you a good explanation for when they decide you get a new one and how many changes decides you get a new one, and someone decides all that. Yeah. So yeah, so this is, so like that's shown, where is that number 493? Right, it's not on there because it reverted. But so basically in BA2 and BA3, the Q had turned into an R, but this is looking at the mutation profile for 4 and 5, like on this particular graph. So you won't see that reversion on this graph because it reverted to wild type, and though you'd write that as without this changed amino acid at the end because it went back to its original. So BA4 and BA5 have a reversion at position 493 compared to like BA2. Yeah. Like insertions? That's actually a really good question. I don't think I've ever seen in literature when there's an addition. I've only ever seen like point mutations and deletions. There must be a symbol for it, but I don't know. Sorry, I can look that up and let you know. Yeah.
So, showing BA5 and BA12, like L to R.
Yeah, so that's in BA4 and BA5 as compared to like BA2.
So like the L would have been in BA4, but it's R in BA5.
So it's R in BA4 and 5. It was L in BA2. Okay, cool. Yeah. Any other questions about mutations and terminology? Everybody ready for their exam on Thursday? Seeing some looks, yeah. It's a mix, yeah. But you can answer in whatever order you want. I'll just give you a packet of paper. No, they're all like shuffled. So you might see chapter 5 first, and then chapter 3, and then back to chapter 5, or whatever. It's just in random order, yeah. Is it what? Oh, I don't know. I mean, I tried to sort of balance concepts from everything we learned. I don't I didn't like count how many questions I put from each chapter, and the different questions require, different amounts of work. Like multiple choice ones are usually relatively quick in comparison to, the ones where you got to draw something. So, I don't know that I necessarily gave you like an equal number of questions from each concept, but there's, I guess, an equal maybe amount of work from each concept, if that makes sense. Approximately. Did my best. Any other questions about the exam or any concepts anyone wants to go over, or review questions you want to go over with the whole class? Yeah. Yeah, it is not. It is required, in that you have to know how to solve questions like that. It's a whole thing that goes through like a protein purification. So it follows. We did not go over it in like this lecture, but we went over all the concepts from it in this lecture, like all those different types of column chromatography we reviewed last week and all the genetic engineering concepts. It's very similar. It was written by Professor Middleton, who usually teaches this class, and so it adheres really closely to what we have been learning in the class. Yes, and I thought I had written in the description for that file that it was required. I apologize if that did not show on your end. That I tried really hard to make it obvious that it's required. Yeah, it might be me. I don't know. Yeah, I did look through student view and there. I don't know. I did my best, I guess. But there's nothing new in there. Like if you know chapter 5, you can do case study 5. Like definitely review it for yourself, like between now and Thursday. But there shouldn't be a lot of new stuff in there. Any other questions? Yeah.
For the actual case study with the CD77, why was found in the supernatant? I thought was salting out, you've got like the end and there was like whatever bone was still more soluble in it.
So there's 2 different salting out steps. So let me pull up the salting out slides and show you. Here we go. I'm looking at the wrong chapter, aren't I?
Oh yeah.
Just realized that. It's fine. So, yeah, so salting out, generally you have multiple steps. So the first part of salting out, you'll usually precipitate out proteins that are less soluble than your protein of interest using a lower concentration of ammonium sulfate. Then you'll take the supernatant from there, discard the precipitate that's in the pellet. That supernatant, you then add even more ammonium sulfate to it. That will precipitate out your protein of interest into this pellet down here. And so then your supernatant after the second salting out does not contain your protein of interest. The supernatant after your first salting out does contain your protein of interest. Does that answer your question?
Yeah,
Okay.
I just didn't realize that there was 2 steps.
Yeah, it's 2 steps. Yeah, that is important for sure. Any other questions? Okay, well, if no one has anything particular they want to review, I guess we can go a little bit early. If you have something you want to discuss with me individually, I have office hours right after this today. I'm available by appointment tomorrow. Just send me an email. I have office hours extended on Wednesday. It's 1 to 3. And other than that, yeah. We are having lab in person this week. I think you're thinking of like the second half of the HPLC lab. Yeah, the LDH lab is all in person. Yeah. Yeah. So yeah, so make sure you hand in your project topics by tonight and make sure you're ready on Thursday for the exam, and good luck.